Hello everyone, welcome back to our class. From what we learned in the last class, we know that every living cell has a transmembrane potential. When a cell is in a resting state, the potential across its membrane is called the resting potential. So, when a cell is stimulated, will its potential change? What kind of change will occur? An action potential refers to a rapid, long-distance propagable potential change generated by a cell upon receiving an effective stimulus based on its resting potential. Take a nerve cell as an example. When it receives an effective stimulus, its membrane potential gradually depolarizes from -70 MV to a certain potential level. It then rapidly rises to +30 MV, forming the ascending phase of the action potential. Then it quickly drops back to a level close to the resting potential, forming the descending phase of the action potential. Together, these two phases form a spike-like potential change, known as the spike potential. The spike potential is the main component of the action potential, and is regarded as the hallmark of the action potential. The part of the ascending phase of the action potential that is above the zero potential line is called the overshoot. Let's take a look at what changes occur in the cell membrane during the action potential. First, let's talk about the conditions for generating an action potential. Again, take a nerve cell as an example. Not all stimuli can induce an action potential in a nerve cell. For a cell to respond to a stimulus, the stimulus must reach a certain magnitude. This usually involves three parameters: the intensity of the stimulus, the duration of the stimulus, and the stimulus intensity time change rate. The minimum stimulus intensity that can induce an action potential in a cell is called the threshold intensity. A stimulus equal to the threshold intensity is called a threshold stimulus. A stimulus greater than the threshold intensity is called a superthreshold stimulus. And a stimulus less than the threshold intensity is called a subthreshold stimulus. The so-called effective stimulus refers to the threshold stimulus or superthreshold stimulus that can induce an action potential in a cell. When a cell receives an effective stimulus, it first causes some sodium channels with high excitability on the membrane to be activated and open. As a result, a small amount of sodium ions diffuse into the membrane along the concentration gradient and potential gradient. The influx of sodium ions causes depolarization of the cell membrane. When the membrane potential depolarizes to a certain value, it triggers the massive opening of sodium channels. The membrane potential value at this point is called the threshold potential. Depolarization reaching the threshold potential level is a necessary condition for a cell to generate an action potential. When the cell membrane depolarizes to the threshold potential level, a large number of sodium channels open, allowing a large amount of sodium ions to enter the cell, and the negative potential inside the membrane quickly turns into a positive potential. The electric field force of positive inside and negative outside caused by the influx of sodium ions will prevent further influx of sodium ions. When the driving force from the concentration gradient and the electric field force from the potential gradient reach equilibrium, the influx of sodium ions stops, reaching the sodium equilibrium potential. When the ascending phase of the action potential reaches its peak, the sodium channels inactivate and close. At this point, the voltage-gated potassium channels open, and potassium ions rapidly flow out of the cell along the electrochemical gradient. The potential inside the membrane drops rapidly, returning to the resting potential level, forming the descending phase of the action potential. After the action potential, although the membrane potential returns to the resting potential level, the concentration of sodium ions inside the membrane increases, while the concentration of potassium ions outside the membrane increases. This change in ion concentrations inside and outside the cell activates the sodium-potassium pump, The sodium-potassium pump pumps out the sodium ions that entered the cell and pumps in the potassium ions from outside the cell, preparing for the next cell excitation. To summarize, the ascending phase of the action potential is formed by the rapid and massive influx of sodium ions. The descending phase of the action potential is formed by the efflux of potassium ions. The restoration of the uneven ion distribution state requires the activity of the sodium-potassium pump. The action potential has the following characteristics. First, the all-or-none phenomenon. When a threshold or suprathreshold stimulus is applied to a cell, the membrane depolarizes to the threshold potential level, triggering the generation of an action potential. For a single cell, once an action potential is generated, its shape and amplitude remain unchanged and will not continue to increase with the increase in stimulus intensity, which is the all part. If a subthreshold stimulus is applied to the cell, the membrane depolarization cannot reach the threshold potential level, and no action potential is generated, which is the none part. In short, an action potential either is not generated at all, or is generated at its maximum amplitude. Just like when you guys take the driving test, there's only a pass or a fail. Second, non-decremental conduction. After an action potential is generated, it is not limited to the stimulated area, but spreads rapidly along the cell membrane to the surrounding areas until the entire cell sequentially generates the same potential change. During this conduction process, the waveform and amplitude of the action potential remain unchanged. Third, pulsatile nature. After a cell is excited once, its excitability undergoes a series of changes during the absolute refractory period. The cell no longer responds to any strong stimulus. Therefore, multiple action potentials generated by continuous stimulation always have a certain interval and do not overlap, showing a pulsatile pattern. For some patients with nerve damage, in rehabilitation treatment, common therapies include neuromuscular electrical stimulation, functional electrical stimulation, and transcutaneous electrical nerve stimulation. While electrical stimulation stimulates motor nerves and muscles, it also stimulates afferent nerves, which project through the spinal cord to higher centers, promoting functional reconstruction. Therefore, neuromuscular functional electrical stimulation plays a very important role in rehabilitation treatment. We know that only threshold and suprathreshold stimuli can generate action potentials. Now, here's a question. What response does a cell have to a subthreshold stimulus? A single subthreshold stimulus cannot trigger an action potential, but it still has a certain effect on the membrane potential. We call the local depolarization below the threshold potential level caused by a subthreshold stimulus a local excitation or local potential. Compared with action potentials, local potentials have the following characteristics. First, graded potential. That is, its amplitude is related to the intensity of the stimulus, and it does not have the all-or-none characteristic. Second, decremental conduction. Local potentials spread to the surroundings in an electrotonic manner, and their spread range is generally no more than 1 millimeter in radius. Third, summation phenomenon. If continuous stimuli are applied to the same site of the cell membrane, the local potentials generated successively can overlap, which is called temporal summation. If stimuli are applied to adjacent sites at the same time, the local potentials induced can overlap within their electrotonic propagation range, which is called spatial summation. If the local potentials sum up to cause the membrane to depolarize to the threshold potential level, an action potential will be generated. So far, we have finished learning about cellular bioelectricity. Let's summarize the characteristics and generation mechanisms of different types of cellular bioelectricity together. The resting potential is the potential in the resting state. It is mainly generated due to the uneven distribution of ions. There are two main reasons for the uneven distribution of ions. One is the transport by the sodium-potassium pump, and the other is the barrier effect of the cell membrane. Under stimulation, cells can generate action potentials or local potentials. It mainly depends on the intensity of the stimulus. Threshold or suprathreshold stimuli can generate action potentials, while subthreshold stimuli generate local potentials. You can remember the characteristics of these two types of potentials by associating them with each other.